The isoelectric point of a protein or amino acid tells us at what pH is the net charge of that protein or amino acid 0. But before we delve too deeply into the specifics on how to calculate the isoelectric point, it's important to know how to determine the charge of our amino acid. It is important to remember that our amino acids, and thus our proteins, change in their charge and in their overall structure when we change the acidity or alkalinity of the environment. So let us look at glycine, for example. Let us imagine that we have placed glycine in a pH 1 solution and we are performing a titration. When we placed our glycine into the pH 1 solution, structure 1 will be present as glycine is in an incredibly acidic environment. By taking a closer look at structure 1, we can see that our amino group and our carboxyl group are both protonated. So if we observe this structure and count its overall charge, we can see that the amino group is +1. So therefore, our overall charge thus far is +1. Our methyl group just below is neutral in charge, and so is our carboxyl group. Therefore, the overall charge of our glycine molecule is +1. This is the structure of glycine in a low pH environment. Now, as we add some base and increase our pH, we can see that our glycine molecule begins to change, so that the carboxyl group has now been deprotonated. So now if we look at the overall charge of our amino acid, we can see that the amino group is +1, our methyl group is neutral, but now our carboxyl group has a negative charge. Therefore, when we add them all up, we can see that our glycine molecule is now neutral in charge. As we keep increasing the pH, making it more and more basic, our amino acid changes structure one last time, and we can see that the amino group has finally released its proton. This means that our glycine molecule now has a charge of -1. However, there is a key item that we have missed in our explanation. So if we look at structure 1 and structure 2 of glycine, we can see that they are in equilibrium, but it says pK1 next to it. So what does that mean? Now it's important to remember that amino acids are weak acids, meaning that unlike hydrochloric or sulfuric acid, they will not just throw away their hydrogens to anyone and everyone. When these strong acids are placed in water, they will completely dissociate. As for weak acids, they will only partially dissociate, meaning that the weak acid will have a dissociated state and an undissociated state that will coexist in equilibrium. Not only is the pKa a good indication of just how strong our weak acid is, but it also tells us what our molecule will look like in terms of its structure, depending on the pH. Now, to explain this a little bit better, we need to have a look at our Henderson-Hasselbalch equation. So when the concentration of our weak acid and its conjugate base are equal, we see something interesting happen to our equation. What will happen is that log base 10 of 1 equals 0, so the entire part of log base over acid will cancel itself out, meaning that our equation will turn into pH equals pKa. Therefore, the pKa value will tell us at what pH level we will have a 50-50 split of our weak acid in its undissociated state and its dissociated state. So let us come back now to our glycine molecule. Our aim here is to determine the isoelectric point, or the pH in which our amino acid is neutral in charge. So we are wanting to calculate at what pH will our second structure be predominant. Here is where we use our pKa values. So if we look at structure 1 and structure 2, because we know that the pKa of structure 1 and structure 2 is 2.34, we know that glycine at pH of 2.34 will be comprised of 50% of structure 1 and 50% of structure 2. Now the exact same argument can be made for structure 2 and for structure 3. This means that at a pH of 9.6, 50% of our glycine molecules will be in structure 2, and the other 50% will be in structure 3. Because we know that at a pH of 2.34, 50% of our glycine molecules are in the structure 2 configuration, and at a pH of 9.60, 50% of the molecules are in the structure 2 configuration, in order to get the isoelectric point, so in order for us to determine at what pH will structure 2 be the most predominant, we simply take an average of these two pKa values. Therefore, 2.34 plus 9.60 divided by 2 will give us 5.97. Therefore, the estimated pH in which the glycine molecule is predominantly at a neutral charge is at a pH of 5.97. It is important to point out that with glycine it has a neutral R group, so when determining the isoelectric point, we only looked at the carboxyl and the amine group. But what happens if we have an amino acid with an acidic or a basic R group? So if we have a look at glutamate or glutamic acid, we can see that it has 4 different structures, whereas glycine only had 3. So the first thing that we need to do is look at each of our 4 structures, and we need to determine which one has the neutral charge. So if we look at structure 1, we can see that our amine group, which is +1, we have the side group, which is neutral in charge, and the other carboxyl group is also protonated, which means both of them are neutral. Therefore, structure 1 has a total charge of +1. Now structure 2, our carboxyl group has been deprotonated, meaning that the overall charge is now neutral. Structure 3, the carboxyl group has been deprotonated, meaning that the overall charge is now -1. And finally, amine group has been deprotonated, meaning that we now have an overall charge of -2. To determine the isoelectric point of glutamic acid, we need to determine at what pH will structure 2 be dominant. Now, I wish to be very clear here. Please do not take the average pKa of all 4 structures. Instead, what we want to do is we want to take the average of the 2 pKa values on either side of our neutral structure. We can see that the equilibrium, the pKa value, for structure 1 and structure 2 has a pKa of 2.19. So that tells us that at a pH of 2.19, 50% of our molecules is going to be in structure 1, and 50% of the molecules are going to be in structure 2. Now, if we have a look at structure 2 and structure 3, it has a pKa value of 4.25. So what we need to do is take an average of those 2 pKa values. So we're going to take 2.19 plus 4.25 and divide it by 2. And this will give us an isoelectric point of 3.22.